Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is The Predictive Power of Poop, COVID-19 Wastewater Surveillance to Inform Public Health Decisions in Colorado. This webinar is sponsored by Bio-Rad Laboratories. Our speakers today are Dr. Susan DeLong, Associate Professor in the Department of Civil and Environmental Engineering at Colorado State University, Dr. Carol Willis, Professor of Microbiology, Immunology, and Pathology, also at Colorado State University, and Dr. Heather Pitcock, Chief Medical Research Officer, also at Colorado State University. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of the webinar presentation. We will ask all our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will see a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Susan. Please go ahead with the presentation.
Thank you so much, Julia. So I'm going to start us off by framing the motivation for wastewater epidemiology. So wastewater epidemiology is actually an established field, this idea that we can monitor viruses in wastewater to get a sense of the prevalence of a disease in a population. And this has been done previously mostly with polio. Early on in the pandemic, work was done in Europe to look at SARS-CoV-2 in wastewater. And this is a published graph from a study in France, which shows that the levels of SARS-CoV-2 in wastewater track clinical data, in this case, fatal cases in France as well as Paris. You can also see that in red, the untreated sewage samples, the line, that curve there for the levels of SARS-CoV-2 in wastewater, that actually the data, the detection of SARS-CoV-2 in wastewater precedes the clinical cases. So it was demonstrated early on in the pandemic that monitoring wastewater could be an early indicator, giving us 2 to 10 days of advance notice that an outbreak is about to occur in the community, which is really the key motivation for using this tool and the reason the word predictive is in the title of this talk. So the reason that this works is that people who are infected with SARS-CoV-2 will shed the virus in their feces very early on in infection. You can see in this graph the log RNA copies of the genetic material of the SARS-CoV-2 virus as a function of days since onset of symptoms. And early on, when symptoms occur, the fecal shedding is very high, and it does occur prior to symptom onset. And importantly, both pre-symptomatic, so before symptoms set in, as well as asymptomatic, people who never have symptoms of this disease will shed the virus. So we can get an indication of the levels, the general levels of infection in a community without people needing to feel sick, go to the doctor, get a test, wait for that test result to come back. So that's another reason that this is an early indicator and a helpful parameter to monitor for public health decision making. So in this talk today, we're going to talk about 2 use cases for monitoring SARS-CoV-2 or the virus causing COVID-19 in wastewater: large population monitoring and small population monitoring. Large population monitoring involves taking samples at wastewater treatment plants, at the influent of wastewater treatment plants, and this allows us to monitor in entire communities with a relatively small number of samples. So here you can see that we might monitor between 20,000 and 2 million people with wastewater treatment plant samples. We're doing that in the state of Colorado at 20 locations twice a week, and the samples are sent to our testing laboratory at CSU, and the data is used to plot trends in viral copy number and help epidemiologists at the state make decisions about social distancing status. The second use case is small population monitoring. And in this case, we might monitor a single building or a facility. We're doing this on the Colorado State University campus, monitoring dormitories, and I'll talk about our study design there. This is a much larger number of samples because, of course, we're monitoring smaller populations of individuals, and the use case here is a little bit different. We can use this information to direct follow-up testing to try to identify the infected individuals. So I'm going to dive in now to how we actually go about collecting the samples and how we developed our sampling plan. So first I'm going to talk about the monitoring that we're doing at Colorado State University for building level monitoring. So early on, we decided to monitor all of our dormitories and residences, student residences on campus. So in thinking about this building level monitoring strategy, we wanted to think about which locations would be the most strategic and beneficial with respect to controlling outbreaks on campus. And we started with a preference for residences over administrative or other types of buildings on campus. And this is really due to the timing of people's contribution of fecal matter to the sewer. We sort of know that that's generally a morning activity, and because, as I said early on in the talk, this virus is mostly shed in feces and wastewater, we really want to capture that signal to get a sense of the prevalence of infection in our community. So we targeted residential buildings rather than buildings where people work on campus. This also allows us to direct follow-up testing because we can link individuals with the buildings they live in and message those individuals to participate in follow-up testing that will be discussed later in this talk. So once we had decided to monitor dormitories, what we needed to do was an in-depth study of the sewer system at CSU, and this was done in collaboration with engineers that are part of our facilities team at CSU. You can see sort of a grayed out image of a building there, and in very faint green lines there's a map of the sewer system. And so the goal is to find manholes that can be linked with an individual building or a small group of buildings, so that facilitates selection of smaller groups of people for follow-up testing if we detect the virus in the wastewater. There's also a lot of complexities to this decision-making process. We wanted to avoid mixed-use buildings to the extent possible. So some of our buildings have dining commons. That also complicates the contribution to the sewer. We wanted to limit sewer contributions from off-campus buildings so that we knew we were monitoring mostly our students, and we wanted to avoid contributions from unidentifiable contributors. So if a manhole also receives water from the gym or the library, it will complicate determining how to direct follow-up testing. And then there's also other practical considerations that I highlight here. So you can see a picture of one of my team members in Tyvek suit and a fluorescent vest there, and there's a nice manhole on a grassy strip, safe to access for the team. That's an ideal location. Some of the ideal manholes that we identified were actually in the street, and this is just an example from not a location on campus, but we don't want to be sending our student samplers into the middle of the street. This is just a quick picture of the wastewater sampling. This is Jason Graham taking a sample, and that's how it's done at the wastewater treatment plant, so it's much more straightforward. A little bit more about the building level samplers. I wanted to provide a few tips and perspective here. We use composite sampling pumps. These pumps collect samples over a 24-hour period. So rather than a grab sample where you just go out and take a sample at a certain time point, we wanted to collect samples over a 24-hour period to make sure our samples were representative. We use compositing pumps that are hung in the sewer. This keeps them out of sight, safe, and actually prevents them from freezing in the winters because the temperature in the sewer is more moderate. You can see there a picture of one of the strainers in the middle photo that hangs in and collects the wastewater. Our samplers are programmed to collect water every 15 minutes over that 24-hour period. Here's just a few more pictures of the team in the field, learning how to program the samplers and showing how the samplers are hung in the sewer. A few more sample examples of field work. I just like to highlight here the importance of safety training, that our team is trained in ergonomics to make sure that, there are no injuries associated with lifting and lowering these heavy samplers, slips, trips, and falls, biosafety hazards. There are a lot of considerations in deploying a team to do this kind of sampling to ensure that it can be done safely. So once we've collected the sample from a community or a building, and we are taking a 24-hour composite sample, those samples go over to the lab where we have a protocol to remove solids, concentrate the virus, extract viral RNA, and do the quantification, which our next speaker will discuss in detail. I wanted to talk a little bit about some of the sample collection challenges as they relate to the downstream analysis. So some things to make everyone aware of. Flow rates vary in sewers over time, and the flow rates can be very low. The composite samplers that we purchased are off-the-shelf commercial samplers, and they're not really designed for building level sampling. And so we had to take into some considerations low flow, and I'll show you some modifications in a moment. It's also worth noting the complexities associated with sewer sheds as well as buildings. We're really interested in black water that comes from the toilet, and sewer sheds have all different contributions. Kitchen waste at dorms and dining commons, laundry, strong cleaning agents can cause issues. And then if we're looking at a sewer shed, we might have contributions from industrial wastewater. And then another challenge is variation in the viral extraction efficiency. So I want to talk about some strategies that we used to address these challenges. And another one that's not noted on the slide here is for building level samples, there's a lack of homogenization. So the samples are not super well mixed. So we need ways to account for that. So we developed some strategies within the sampling framework to innovate on strainer design, and then we have controls that can be used to measure concentrations of fecal matter, which helps address the issue of various inputs beyond just black water from residences, as well as the lack of homogenization. And then we have a control that helps us to track the percent recovery for every sample that we analyze. So just some quick photos of strainer designs. We had to develop new strainers that had a lower diameter. So a 1/4 inch strainer will work much better for building level sampling because the flow is so low, the depth of the water is low. In the middle, you can see a strainer that's designed to limit clogging. We've definitely had challenges with issues of various things that end up in sewers clogging our strainers. And then on the right is a low flow strainer, which only has holes on the bottom, and that little wing plate helps it to not roll in the sewer, and this can work in really low flow situations. Here's a slide that presents some of the controls that we're using. We have used 2 different endogenous viruses as a control for the level of fecal matter, which can be used to normalize. F+ bacteriophage is associated with gut microbiome, and so it's an endogenous virus associated with fecal matter. This has worked really well for our state samples. We've also used the Pepper mild model virus, which is another indicator of fecal matter. We can track those 2 viruses to help us understand the level of fecal material in a particular sample, as opposed to other contributions, wastewater contributions like laundry water. And then for our percent recovery spike, we really wanted to use a coronavirus. We wanted a virus that had the same surface chemistry and would behave in a similar way in our extraction protocol to SARS-CoV-2. And for that, we were able to use this bovine coronavirus vaccine. And we spiked that into all of our samples prior to extraction of the virus, the SARS-CoV-2 virus for analysis. And so these are some of the strategies we've used to address the complexities that originate in the sewer system and on the sampling side. And with that, I'm going to turn it over to Dr. Carol Willis to talk about the analysis part.
Thank you, Susan. So when the lab receives the wastewater samples, there are a number of things that we need to think about when we were developing the assay in order to measure the levels of the coronavirus in it. There are some good things about working with sewage, believe it or not. One of the main advantages is there's plenty of it, and that made it quite easy for us during the summer to access large quantities to develop our assay, and the city of Fort Collins provided us with a lot of sewage samples over the summer. Another advantage is that it's non-invasive and it requires no IRB approval to take these samples because we can't track anything back to the individual that contributed. As Susan mentioned, it gives us a nice snapshot of the population, And of course, you can't underestimate the power of poop jokes, especially for those working in the lab. The cons of working with wastewater are there though, and we do take those very seriously. For example, coronavirus is not the only virus that's present in wastewater. There are a lot of very nasty viruses, hepatitis A, rotavirus, norovirus, and these viruses are specifically designed to transmit through feces, and so the viruses that are present are actually still infectious. So we have to handle our samples pretty carefully. Another disadvantage is that the samples are pretty dilute. There's a lot of water going through the sewers, obviously, and so that we need 40 mils of sample to get enough virus to detect. So we have to concentrate that because we can't deal with a 40 mil sample and just put it into a PCR reaction. Wastewater, of course, has a lot of yucky stuff in it, and these chemicals and molecules can inhibit our reactions that are designed to detect the virus. And so we need to account for that. And as Susan mentioned, the samples aren't homogeneous, which means that we do get a lot of variation from one sample to the next. And from one day to the next, and between samples from different locations. And all of that means that although our experiments have run very carefully, there is a lot of noise in this type of data. So Susan mentioned the steps that we go through. Basically, we spike in a recovery virus, we separate out the particulate matter, we concentrate the virus, we extract RNA, and then we quantify using a PCR approach. There were a number of things that went into consideration when we were developing our approach for quantifying the virus. We had a list of qualities that we wanted our assay to have. The first one here, being fast, was really important because if we're trying to predict what's going to happen in the future, we can't wait 2 weeks to turn around our results. We needed a 24-hour turnaround from receipt of the samples in the lab to when we're producing the data. We wanted the assay to be pretty easy. This is because we did not want to have to go find highly trained technicians in the middle of a pandemic. We needed to be able to hire somebody that was basically competent in molecular biology, needed to be able to run these assays. As I mentioned, it's important that the assay was resistant to inhibition, and we wanted it to be sensitive so that we could detect the virus as soon as it appeared, and accurate so that we could quantify it. Unlike with a clinical test where you just need to know whether the person is positive or negative, we actually need an accurate quantification of the number of copies of the virus in each sample. Ideally, we wanted it to be cheap to set up and run these assays, and it turned out to be really important that the supply chain for all the reagents and supplies we needed to run the assay needed to be robust. And this has been a real problem with a lot of the clinical diagnostic testing. The supplies for real-time PCR have sometimes been hard to come by, and we've managed to avoid most of those issues. And finally, we wanted to be able to rely on our existing equipment and expertise rather than having to buy a lot of new equipment and find additional people to perform the experiments. So for the clarification and concentration steps, the clarification, getting rid of the solids, we've used filtration and centrifugation. These are both fast and easy and cheap. Both work equally well. So these have been our go-to methods for just getting rid of the lumps that are in the sample. And then for the concentration, we decided to go with an InnovaPrep concentrating pipette, which was recommended to us by a wastewater treatment facility who were using this approach already. This pipette is really neat. Basically, we stick this probe into our sample. It sucks up the 40 mils of liquid, and then we elute it off of the filter into 400 microliters, and this takes 2 minutes. So we basically take 40 mils of wastewater and we turn it into 400 microliters, and we'll also get rid of some of the other smaller, really small particles in a 2-minute period. And so this is the reasons we picked this is that not only does it work really well, it's fast and it's really easy to use. This instrument it tells you how to use it every step. For the RNA extraction, we tried Trizol extraction and the QIAGEN prep. In the end, we went with the QIAGEN kit, primarily because it was easy. There were less problems with the DNA not being able to be amplified or RNA not being able to be amplified afterwards. But both of these approaches work pretty well. Trizol has the advantage of being cheaper, and the supply chain is more robust, but we haven't had any trouble with the supply chain for the other reagents either. So for the quantification step, this is probably the most important step for us. This is what gives us the data. We were very fortunate in that we already had on site a Bio-Rad QX200 digital droplet PCR system. This had been purchased in 2015. It had been in use on campus in my molecular quantification core for 5 years. My lab was very experienced in using this. They've been using it for measuring gene expression in various samples for a while. So that was one real strength of our approach, was we already had this equipment available. But we also focused on this because we do have real-time machines available as well. But we decided to go with digital PCR also because there's less demand for the reagents, and the supply chain has been robust. We've had no problems getting the supplies we need to run the assays. Digital droplet PCR is particularly resistant to inhibition, which is a big advantage that it has over real-time PCR. It doesn't really get affected by all of the yucky stuff that might come through with the RNA from the wastewater. And then another real advantage is that it gives absolute quantification. When we put the RNA into the sample, the data that we get out at the end tells us the exact copy number of virus in that specific sample without any need for a standard curve. And that's because we're actually reporting copy numbers and not just positive and negative. This was actually really helpful. Digital PCR is also more sensitive than real-time PCR. So we were able to detect the virus at lower levels than you would do with real-time PCR, and it's also very accurate. And finally, there are reagents available that allowed us to perform the reverse transcription, turning the RNA into DNA in the same step as the PCR reaction. So that saved us some pipetting and some time, and allowed us to turn around our data more quickly. So this is an overview of the process that we go through after we've isolated the RNA from the wastewater. The reactions are just regular PCR reactions with a fluorescent probe. They get set up in individual tubes. So each sample has 2 reactions set up for it, one for the coronaviruses and one for the control viruses. And then each of those reactions gets partitioned into 20,000 droplets. And so droplets sometimes get a copy of the RNA, and sometimes they don't get a copy of the RNA. And this is just done by mixing it with oil in a droplet generator. And so then these individual droplets go through thermocycling, cycles of heating and cooling that allows the DNA to be amplified. And at the end of the reaction, any droplet that received a copy of the viral RNA will have a fluorescence will have a fluorescent signal, whereas droplets that didn't receive a copy of the viral RNA will have no fluorescence. And we put the reaction through a droplet reader, which is essentially like a flow cytometer for those of you who know about flow cytometry. And that looks at each droplet and measures the amount of fluorescence in two different channels, a red channel, a green channel and a blue channel. And from that amount of fluorescence, we can count the number of positive droplets, and using a mathematical equation connected to the Poisson distribution, from that, the software that is provided with the machine can determine the exact copy number of the virus in each sample. And so this is what the data looks like. On the right, you can see there's the gray band at the bottom is the negative droplets, and then the blue and the green are the positive droplets in two different channels. So the green channel is in this particular reaction is the bovine coronavirus, and the blue channel is showing the SARS coronavirus probes. And so you can see that the SARS coronavirus probes are labeled with a different fluorescent molecule, and so they fluoresce in a different channel, and we can detect them separately even though they're in the same tube. One thing to notice is that the amount of the positive blue droplets varies across the different samples, so that each column there is a different wastewater treatment plant or manhole, and you can see the blue droplet number will vary, and that's because the amount of coronavirus varies from in different locations. Whereas at the bottom, the bovine coronavirus, you can see that the number of green droplets is pretty similar across the board. Although one or two of them might have lower numbers of droplets. This is because we put the same amount of bovine coronavirus into each reaction. We know how much is there. And so we should get similar concentrations of bovine coronavirus across the board, although sometimes if our recovery is bad, for example, if there are chemicals in the wastewater that affect the survival of the virus, then we can detect that using the bovine coronavirus control. So we perform a reaction that contains the coronaviruses, and a second reaction to detect the endogenous controls, the coliphage and the Pepper mild mosaic virus. Okay. So Susan already talked a little bit about the controls, so I won't go into them in a huge amount of detail, but what I wanted to point out was on the right here, I'm showing the copies per liter of the F+ and the Pepper mild mosaic virus. And you can see that the F+, for example, in the UG lane, in the UG sample, can be pretty low, and sometimes we don't detect it at all. And we think that that's because in that particular location, there are chemicals or something going into the water that are affecting the survival of that virus and our ability to detect it. In this case, the Pepper mild mottled virus is the signal from that is more consistent. The other thing to notice is that, especially for the Pepper mild mottled virus, the signal is pretty consistent across the board with for different sites, whereas the F+ seems to be a little bit more variable. But we found that F+ is great for our state samples, and the Pepper mild mottled virus seems to be working a little better for the dorm samples. And we also do controls for contamination, so a no template control where we don't put any sample in. We should never get any signal, so that's just to make sure that all of our reactions are clean and we're not mixing between different reactions. And then we do positive controls with inactivated virus just to make sure that all of our reactions are working. So this is just an overview of the whole process. So we take our wastewater that comes into the lab, we add spike of the bovine coronavirus vaccine, we filter or centrifuge, we ultrafiltrate to concentrate the virus, we extract RNA, we assay in 2 separate reactions to detect 4 different viruses using digital PCR, and then at the end we get out these droplet numbers that will give us an indication of the levels of virus in the sample. Our limit of detection is about 3,000 to 5,000 copies per liter. The limit of quantification is about 13,000 to 15,000 copies per liter. And all of these criteria get reported on a dashboard, either to the CSU Rapid Response Team, which Heather will be telling you about in a minute, or to the Colorado Department of Public Health and the various wastewater treatment facilities. And that this data gets uploaded within 24 hours of us receiving the sample. almost all of this, except for the data analysis, is done in one day. Pretty much the people in the lab will put the PCR reactions on to run before they go home at about 9 or 10 o'clock at night, and then they'll either come back and read them later, or I will go in and read them in the morning, and then I analyze the data, or a graduate student will analyze the data first thing in the morning. And so we are able to get through this whole process within 24 hours of receiving the samples, and report those results back within 24 hours. So as fast or faster than a clinical assay, which only tests one person, whereas we're testing hundreds to thousands of people. I'm going to tell you a little bit more about what we do with the data in a minute, but before I go on to that, I want to talk first about variant detection. So probably you're aware that there are a large number of variants or a number of variants of concern arising around the globe. And these have different properties than the original coronavirus. Some of these properties are particularly concerning. A mutation in the spike gene, N501Y, is seen in the UK, South African, and Brazilian variants, and it's been shown to help the virus spread more easily. And then, on the other hand, the E484K mutation is seen in South African, Brazilian, and in some UK variants, and that may affect the ability of the body to recognize that virus after it's been vaccinated. So the antibody response to the wild type virus may not recognize the E484 virus. And so these variants are concerning because we cannot they have different properties, and so all of the mathematical models and our responses are based on the wild type virus, and these new viruses have different properties, and so we need to adjust our models as these variants appear. And we also we possibly will need to be more aggressive with our quarantine and other responses if these become widespread. And so we want to be able to detect these in wastewater, and we've been able to show that this is possible. This on the right here, top right, the N1 is the regular primer probe set that we use to detect the coronavirus. And then on the left, N501Y is detected by an assay developed by Bio-Rad that has two different color probes. One, the blue fluorescent probe detects the mutant virus, so the N501Y variant, and the green probe detects the wild type virus. And you can see in the data on the bottom, it's not all that clearly labeled, but you can see that the blue virus is only present in one of the 5 samples on the left, and whereas the wild type is present in all of the 5 samples on the left, and the samples on the right are just controls to make sure that we're detecting what we say we're detecting. So that 5th sample, the 5th sample from the left, which is right in the middle, we sent that for sequencing, and we were able to demonstrate that the presence of multiple variants consistent with it being the UK variant were present in that particular sample. And so based on this evidence, we're pretty confident that the UK variant was present in that sample, and we can move on to plan accordingly for seeing probably more of that variant as time proceeds. Okay, so what do we do with the data once we have it? As Susan mentioned at the beginning, we're monitoring large populations and multiple wastewater treatment plants across the state of Colorado, and small populations, as in the dorms at CSU, which Heather's going to tell you about more soon. And so with the large populations, what we're looking at is trends in viral copy number, and the data that we're providing is informing public health decisions about social distancing and health systems and the economy, along with it's being used in conjunction with all of the information about clinical case numbers. So we started receiving wastewater samples at the beginning of August, and we've been testing twice a week since then. Multiple wastewater treatment plants across the state of Colorado are sending us their samples twice a week. And this is what the data that we provide looks like. So in the black line here is our data over time. And then the blue line is the clinical cases. And you can see the clinical caseload tracks nicely with the number of copies of virus that we're detecting in the wastewater. And this is just 2 different locations. But importantly, the actual correlation between the amounts of virus in the wastewater and the clinical cases is really quite compelling. And so an R-squared value closer to 1, a perfect R-squared value is a perfect correlation is indicated by this number being 1. And so you can see for this particular wastewater treatment plant sample, we're getting a really good correlation between the clinical cases in orange and the signal that we're receiving in the wastewater in blue. And even for those wastewater treatment plants with less correlation, the correlation is still very good and has predictive value. So the number is not quite so good, but it's still definitely giving us a really useful information. And so when we provide the copy numbers of the virus, it's being taken into account and informing the public health decisions down the road. On the state level for municipalities and for local areas. And so I'm going to stop now because that's the bit that I've been most connected with, and I'm going to hand off to Heather, who's going to tell you about the response that we have at the university level to the data that we're providing.
Thanks so much, Carol. So I'm going to shift the focus to the small population monitoring and talk a little bit about why we chose to do this and how we analyzed the data and responded to it and what the impact has been. So the why of doing it is early identification of asymptomatic people. So the hope was that by identifying them and putting them into isolation early and then putting their close contacts into quarantine early, that we could not only decrease the number of cases, but also decrease transmission to people who might have more severe symptoms and be at higher risk. There are challenges, and these have been touched on before, but in terms of predicting how many infected individuals are contributing to a sample. So some of the variability that Dr. DeLong spoke about in terms of homogenization of the sample. There's also individual variability between the people who are infected, because fecal shedding early infection has not been fully characterized, and there is variability from one person to the next. The relationship between fecal shedding and respiratory infectivity in terms of knowing that timing is not fully known. And fecal shedding can persist even after the quarantine or isolation period is over and that person has returned to their assigned housing. And so early on in the semester, it was fairly easy to say that if we saw a signal, that was an active infection. But as the semester went on, we started having to make determinations about whether the signal we were seeing was due to people returning to housing and no longer being infected, but still shedding fecal matter, or whether it represented new infections that needed to be identified and those people removed from general circulation. So there was, again, variation in flow rate, chemical composition, all the things that have been discussed. So we had some significant constraints as we contemplated bringing this technology online. There was no roadmap to follow, and there still is no roadmap. We didn't even have publications at that point about micromonitoring approaches. There were good publications about testing at the treatment facility level, but we didn't know whether that could be applied to these smaller settings. We knew what the limited detection was, but we didn't know what the threshold for action should be and at what level we might be able to find cases, or, and because the timing wasn't clear, we didn't know if we would see wastewater signals that then could not be identified in the people just because they had not yet had virus in their nasal passages, and thus the nasal swabs wouldn't show anything. we didn't know how long the virus would stick around for, and what to do if we couldn't find the infection. So all in all, we really didn't know if this was going to be a successful and productive thing to do. But we didn't have a lot of other options early in the pandemic. So in the fall of 2020, we just decided that this was something that we had to do, and we had to make decisions based on little to no information. So we decided to start with the lowest threshold possible, which was that limit of detection between 3,000 to 5,000, and then calibrate it to our results. And this definitely was more art than science, particularly later on when we started having people who are no longer infective contributing to the signal. We looked for trends. We found that resolving or increasing trends were much more useful than any individual measurement. And we accepted that occasionally there were signals that resolved and we never identified a case. And we didn't know if that was because the person was a visitor to one of the dormitories, a worker who left and didn't get tested, or what the reason was. And so trial and error was definitely an element in how we did this. As Dr. Willis mentioned, we formed a rapid response team in order to optimize outcomes. And there was a very large degree of expertise that was represented by that group. So we had clinical, scientific, technical, and statistical and economic experts all contributing their help to determining what did the data mean. Then we had legal and regulatory experts helping us understand what the boundaries were of what we could and couldn't do about it. And then we just had a lot of people on the operational side, the student affairs side, Greek life, housing and dining, communications, helping us to understand how to effectively communicate strategies, incentivize students, faculty, and staff to be compliant and help us keep the campus healthy and safe. So CSU worked on developing multiple capabilities, and ddPCR was a big tool for all, or I should say most of the efforts that we launched. So the wastewater testing, you've heard in detail. We also developed a pooled saliva assay, which I'll tell you more about, which was very helpful in being able to expand the number of people that we could screen in a time-efficient and cost-efficient manner. And then we were also outsourcing molecular testing to a CLIA-certified lab that was using an FDA-authorized test in order to confirm the results. So currently, the way that wastewater monitoring is occurring at CSU is that Dr. Willis sends us results letting us know that wastewater was positive for a SARS-CoV-2 signal. The residents of the facility where that signal is located are notified to go and get tested early. So now everyone who has a campus presence must be tested, but they're asked to test early in the week. And then, again, that's analyzed by digital droplet PCR, first in a pooled approach, and then by our outsourced commercial partner who is doing the authorized testing. So, for example, if we found that there were 6 positives within a building after doing that screening, we would then look again to that wastewater result, trying to make sure that it's coming back down once those positives have been moved into quarantine or isolation. And if it doesn't come down, then we continue to notify those residents and make sure that they do get tested. And at the same time, we look for an increasing result in the quarantine / isolation facility to make sure that we're seeing the movement of that signal. So as I mentioned, small population monitoring is much more about overall trends than about any single result. And what we've seen is it's very reliable if we see multiple increasing signals or we see multiple decreasing signals. Where we don't always find a result that's commensurate with the size of the signal is when we have like a single really large signal. We've almost always find cases when that's the case, but we don't know, we can't correlate it to the number of people that we actually find. So we have multiple different people that are contributing to the response to wastewater monitoring, and all of them are very integral to this process. In terms of the saliva testing, it is a pooled approach where pools of 8 are pulled together and tested. It's done at a socially distanced facility. There's been an aerosol evaluation of that facility, and because it's a gym with very high ceilings and a large volume of distribution, it has been found to be similar to being physically distanced by 6 feet apart outdoors. On average, the process takes 7 minutes, so it's very, very quick. People don't have to stay around in that building for long periods of time. And we only allow asymptomatic people to go get the saliva screening. Anybody who has symptoms has to go directly to get the FDA-authorized CLIA-certified nasal swab. What we find is there is a degree of correlation between the signals that we're finding and the actual cases that we're finding. It's nowhere near that tight correlation that is possible when you're doing it with a large population at a treatment facility. But the flip side of that is because it's a small population, it's much easier to actually have an actionable item as an outcome of the result. So we found that we've had a very high degree of return on investment for this project and for the overall project. The wastewater monitoring has been very helpful in maintaining the health of the residence facilities where people are living in very tight quarters, sharing bathrooms, sharing eating spaces, et cetera. It's really contributed to keeping the campus open to in-person classes through the goal date in the fall of November 20th, which was a big achievement. And we have ample evidence that asymptomatic cases have been identified and proactively put into isolation so that we were able to reduce the overall number of cases on campus. Saliva screening really increased the number of people that we could effectively screen. We were able to notify individuals who contributed to a positive sample because the process allows for identification of the individual and tell that person that they needed an FDA authorized test. So that turned out to be a huge cost-saving measure because we're eliminating the expensive FDA authorized test for people who are negative and only sending people who are very likely to be positive to get those tests. So the cost of analysis was reduced by 86%. Because of the sensitivity of ddPCR, the pools of 8 work very well without substantially increasing the sensitivity of the assay overall. And CSU was able to move to screening anyone who has a campus presence, not just the people in residences, in the spring semester. And finally, the cost per positive test or the cost per case of COVID-19 was reduced by approximately 87%, which is a huge savings. So in summary, asymptomatic, pre-symptomatic, and symptomatic people are all shedding virus that is detectable in wastewater. ddPCR can be used to detect it, and this is partially due to its sensitivity. And SARS-CoV-2 levels can be followed in wastewater. They correlate well with infection rates in very large populations. However, due to the large number of unknowns and variability, which have more of an impact in a smaller population, it's really not possible to directly correlate levels in wastewater with numbers of infected individuals. Trends over time an absence of detectable SARS-CoV-2 in small populations can aid assessment regarding need and timing of individual testing. And so we had some very important keys to success that we were able to identify as a result of all of these projects. And one is that early buy-in from stakeholders that included seed money and in-kind donations were really important to the success of getting these projects started in a timely way that allowed the university to have mitigations in place right at the start of the fall semester. The availability of relevant expertise was really important in understanding what we were finding. Partnerships of public, private, and academic institutions were key. Exchange of information was really important. Prior investment in droplet digital PCR technology allowed the campus to be poised to get these projects up and running quickly. And the robust supply chain for ddPCR reagents was really important to be able to maintain the project when there was a lot of competition for other types of PCR supplies. And then, we depended on free and rapid sharing of information and resources by others, and we tried to pass it on for those who came to us to ask questions about how to do this. And finally, the shared interests and motivations, not only within our own community, but with public, private, and academic institutions and other communities was very important to the success. Any project like this involves the work of just a countless number of people, and this is not even, the full list of everyone, but we want to acknowledge everyone who contributed to the success of this project. So thank you.
Great, yeah, thank you very much, Heather, and thank you to all three of you for this really nice presentation. As a quick reminder, if you have a question, type it into the Q&A panel. And we would also ask our participants to take a brief moment after the webinar has ended to take our exit survey and give us your feedback. Let's see, we had a ton of questions. We will certainly not get to all of them, but let me see what we could address in the time that we have. There were several questions around the procedure that you had after finding positives in a certain building, in a dorm. What was your next step? Would you test everybody in the dorm? Would you shut the dorm down? What was the protocol?
So we only shut 2 dorms down once, and that was in response to that first time we had those extremely large signals because we didn't know what they meant. So the procedure was that if we saw a signal in a dorm, we would ask the residents to go get tested. And housing and dining was wonderful in helping to track the people as they tested, to help assist with the people who were in quarantine or isolation by providing food, transportation, support, and then we would look at the signal, and if the signal decreased substantially or resolved, we would assume that we had found the positives that were to be found, and we started again the next time that we had data. If the signal was not resolving, we assumed that there were some members who had not been compliant that still were infected, and so housing and dining would go back and tell them that they needed to go into quarantine. Our last resort was that if they did not get tested, they would be transferred to quarantine, but we never actually had to do that because as soon as people were told to start packing, they found that they had time to get tested after all.
Okay, great. Yeah, thank you. You mentioned that the wastewater testing did not require an IRB. Did the saliva screening require one?
So the saliva screening is not a research study, so it doesn't require IRB. We did, just like with the wastewater testing, in order to do our due diligence, we did send a question to the IRB and say, this is a public health activity where, if we were to ever publish the results, we would we would get it through the IRB. But as a public health activity, our understanding is that we need consent from people, but we don't need IRB review. And do you agree? And they did agree with that. We did send the consent to the Office of General Counsel for the university to make sure that we were appropriately addressing all the needs, both legal, and we had a regulatory expert to help us also review that consent to make sure that it was within the standards. But everyone who contributed a sample did consent.
Mm-hmm. Okay, great. How sensitive was the wastewater assay? I mean, could you get down to a single infected individual? Do you have any idea how sensitive it was?
I can take that one. So we know we can reliably detect 3,000 to 5,000, so that copies per liter. My feeling is that in a small population like a dorm, I think probably one we would be able to detect them, but they have to poop at the right time. So we don't have continuous sampling. If the person is pooping between the times that we sample, we wouldn't necessarily catch them. And that's one of the reasons we're sampling 3 times a week, because whoever is shedding into their feces will be doing that for more than one day. And so if we miss them one day, we should get them the next day. So we can't guarantee that we would detect one person, but we feel that if that one person did poop at the right time, we should be able to catch them.
Okay. We also had quite a large number of very detailed technical questions. Maybe I can just ask a couple. How much initial volume did you use for the wastewater analysis? Is it only 40 milliliters from 1 liter of entire sample?
So the entire sample, I think, is like gallons. Like it's not. We take I think it's 85 mils every 15 minutes. So we have a very big sample. And from that they shake it up, and then we get 3 tubes of 40 mils each. We process one. So if one 40 mil sample goes into goes through all of the processing, and then one goes into the freezer, and we keep one as backup and throw that away if we don't need it.
Okay. And somebody wants to know whether you've seen any effect of pasteurization of the wastewater, I don't know if you did that, on the ability to detect the SARS-CoV-2 virus by digital droplet PCR.
So initially, the protocol that we started with did involve heating the sample before it was used. We decided against doing that one because we were worried it was going to hurt the virus, and another because it just took an hour, which is another hour onto our processing time. But in the saliva processing lab, I know they do heat for an hour to 70 degrees, and they still have no problems detecting it. And I have read that there's not a huge effect of the pasteurization process, and it does kill all the other nasty viruses. So if you didn't have a biological safety cabinet, maybe that would be one way to get around the risk of handling this material. I think it would be okay to heat it.
Okay. Maybe one more general question. Do you think once the pandemic is over, this kind of wastewater analysis could still be useful to detect other types of outbreaks, for example?
I can take that one. I think it already has been used for other types of infectious diseases, as Dr. DeLong mentioned. And we are very interested in looking into a sort of not just other outbreaks, but even a multiple pathogen detection approach. We think that could be a really interesting strategy to pursue.
Okay, great. And are you going to publish your data, or have you already published it in any way, like a preprint or something?
Carol, did you want to take that one?
We're working on publishing parts of it. We did develop the assay in collaboration with a company, which makes some of the publishing parts are more difficult, but we hope to publish some of it. And if you have specific questions, you're welcome to contact us directly.
Yeah.
The other thing that we've been considering is potentially publishing the entire approach that the university has taken, just because we did feel like it was quite successful in keeping the campus open to our target date.
Okay, great. Thank you. I'm sorry we have actually run out of time, even though we still have a lot of questions. So I'm hoping that somebody will be able to follow up with you via email if we did not answer your question today. So let me thank our speakers, Susan DeLong, Carol Willis, and Heather Pitcock, and our sponsor, Bio-Rad Laboratories. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version via email. So with that, thank you very much for attending this Genome webinar.